Gans. Tonight, I'd like to talk about the origin of life. Imagine a universe devoid of anything alive. There are galaxies, there are stars, there are planets. There are even planets with oceans and blue skies with puffy white clouds, but nothing is alive at all, anywhere in the universe. And then, out of nowhere, suddenly, there's something alive. And the question is, what happened? One moment, one fraction of a second, it was a universe with just lifeless chemicals. And the next second, something is alive somewhere in the universe. How did it happen? How were the first structures of life formed? Scientists don't have a clue. Now, this is the introduction that I received from Rabbi Moshe Averick at a discovery seminar in Chicago a number of years ago. He said to me, why worry about evolution? That talks about what happened afterward, how species evolved, so to speak. But how did the ball start rolling? What happened to begin it all? He says, that's the biggest mystery of all. He gave me a copy of his book, Nonsense at a High Order. And in that book, he quoted several scientists as saying the spontaneous genesis of life is so unlikely as to be close to a miracle. So, for example, the physicist Paul Davies, in his book, The Fifth Miracle, and I quote, "The living cell is the most complex system of its size known to mankind. How did something so immensely complicated, so finessed, so exquisitely clever come into being all on its own?" Now, maybe you remember when you first learned about these things, at least people my age, a long time ago, when the teacher would talk about a cell, they would draw a circle on the blackboard instead of whiteboard, go back far enough, and then a dot in the middle. The circle was the cell membrane, everything inside was the cell, and then the dot was the nucleus, and that was it. A very simple thing, a circle and a dot. Well, a cell is really much more complicated than that, as we've discovered with the invention of more and more powerful microscopes, even though it's extremely tiny, like 25,000th of an inch. It is an immensely complicated factory that produces everything that the body needs to replace itself, to heal. It has a blueprint of the entire body all inside of it. Now, if I were to start listing all the amazing things about the cell, we would be here a whole night. So I'm just going to give you 13, okay? 13 things just to illustrate a little tiny bit of the complexity of this incredibly small factory of which all living organisms are made of. First of all, let's talk about that circle, the cell membrane. You see, things have to get in and out of the cell, right? Fuel, supplies has to get into the cell so that it can keep on burning and working and doing things. Manufactured products have got to get out of the cell or it doesn't do any good. Waste products have to get out of the cell. How do they get through that line, that circle? It's not a circle. You see, it's a very complicated structure. And it has portals, automatic portals in it all over for both input and output. So, for example, it has sliding doors. It has revolving doors. It has suction cups, suction pumps. And it has, at each one of these, substance recognition detectors, things that are going to detect what is this that's trying to get into the cell, or what is this that's trying to get out of the cell. Am I going to let it in, or am I not, or out, as the case may be? So it can identify all the things that are going on, trying to get into the cell, things that are going on around the cell. So, for example, substance recognition detectors, G protein receptors, B cell receptors, T cell receptors, ion channel linked receptors, surface recognition receptors. These, by the way, these surface recognition receptors that will detect if there's another cell nearby. And if it doesn't feel the other cell where there should be, it knows there must be a wound and it starts proliferating there. And that's how a wound clears up. It gets healed. That was number one. Number two, there's a monorail transportation system that runs on rechargeable batteries, all inside that tiny little cell. And it brings things from one end of the cell to the other. If this thing is manufactured here and it's got to get there, it gets onto the monorail, and there's a thing that walks along the monorail carrying its cargo wherever it's supposed to go. These are called microtubules. The rechargeable batteries are called ATP. Then the fuel has to be imported and processed in order to recharge the batteries. Those are done by the lysosomes. That's number 3. Number 4, there are battery recharging stations. That's called the mitochondria. And then there's a high security blueprint room. That is the nucleus. That was that little dot. It's really a whole new structure inside the cell. And it has in it the blueprint of the entire organism. It's called the DNA or the genome. That's number 6, an entire blueprint of everything. Then there's a copying machine. That's number 7. It's called RNA polymerase. And the copy that it makes of the DNA so that it can be copied and used to make whatever product is necessary, that's called messenger RNA. And then there are high security portals into the nucleus. So things from the cell that are inside the cell but outside of the nucleus can't get into the nucleus unless they're supposed to get into the nucleus. And so there's a nuclear pore complex. It consists of 456 proteins, and everything that needs to get in has got a security code. Think about it. Think about the doctor at the hospital, right? He holds his card up by the detector on the door, or he slides his card through the detector on the door, and then it gives him entry. The same thing over here. Anything wants to get into the nucleus, or out for that matter, has got to have the right code. And if it doesn't have the right code, it's not getting in. The portal is not going to open up for him. And the portal itself is incredibly complex. It consists of almost like a circle that closes up like the lens of a camera, the cover of a camera. It goes up from all sides. When something is authenticated, yes, this can get in, it opens up and there's a basket. The thing goes into the basket and it closes again so nothing else can go in. And then the basket goes down through a little elevator, comes out on the other side where another door opens to let it out. That's high security. Then there's the production room, number 9. Production room is called the rough endoplasmic reticulum. And over there, whatever needs to be manufactured with this copy of the DNA is going to be manufactured. But of course, the raw materials have to be transported to the production room. That's done by tRNA. And 11, there are assembly machines that actually make the products, the structures that are needed by the body, and that are going to get out of the cell and go to wherever they're supposed to go. These assembly machines are called the ribosomes. Number 12, there's a final production and a quality control to make sure everything is done absolutely correctly. And that are called the chaperonins. Finally, 13, the packaging and the labeling. It's got to be packaged and it's got to be labeled with an electronic label so that it knows where to go. And also so that it can get out of the cell, because it's going to be examined when it gets to the portals and the membrane. It's going to look at the label on the packaging, say, yes, I know where this is supposed to go. It's supposed to go out, open up, let it out, and then it goes wherever it's supposed to go. And by the way, this is a very, very simplified version of the cell, just to give you a taste of how complicated it is. The question is, how did all of this come together? Right? Everything, all organisms are made of cells. So instead of concentrating on a whole body with a heart and lungs and all that, we're concentrating on the little parts of it, just the cell. Well, there are more than 20 different so-called theories about how this came about. And not a single one of them is a theory. By the way, they all contradict each other too. See, a theory explains how something happened, not where or when. Now, the where and the when are important too, but it's not a theory if it doesn't tell you how it happened. Imagine a detective who was trying to solve a crime, and he comes back to his chief, the police chief, and he says, "Okay, I figured it out. I can tell you when it happened, and I can tell you where it happened." Okay, and who done it, or how did it happen? Well, I don't know that. I mean, that's the most important part, right? So the question is, what is the theory? It's not a theory if it's just telling you, well, I think it happened here, and I think it happened then. Let's take a look at a few examples. One example, warm pond and lightning. Here you have a warm pond, nothing alive, just chemicals in there. The right chemicals, fine, but chemicals, nevertheless, nothing alive. And all of a sudden, there's a stroke of lightning, and lo and behold, out comes Frankenstein. Out comes something alive. Of course, only in the movies, because you know very well that lightning is going to destroy any organic compound that's there. It's not going to make something come out alive except in the movies. So that's so-called theory number one. Theory number two, deep sea thermal vents. Now, in both of these cases, they're telling you where they think it happened, and they're telling you what they think the energy source is, because you see, it requires energy to put together all the little pieces of the cell, right? And that's energy has got to come from somewhere. The problem is that the energy has to be a directed kind of energy. Lightning is not a directed energy. It's just a huge splash of energy in a particular area, but nevertheless, it's not directed. Think of the difference between a lightning stroke and a blowtorch. Okay, a blowtorch is a pinpoint flame that's going to apply the heat in one specific place. Imagine you have a bunch of links that you want to weld together to form a chain. You can do it with a welding torch. You can't do it with a stroke of lightning. The stroke of lightning is likely to just melt the whole thing into a slab. Similarly with the deep sea thermal vents, there's the energy source is the heat from the vents, but it's just heat. It'll put a few molecules together, but then when it gets too complicated, it'll rip them apart again. A third one is radioactive beach. Nice, beautiful beach, except that it's radioactive. Same idea. It happened on a beach with the energy source, radioactivity. Well, we know radioactivity kills. It doesn't make something come alive. Doesn't make someone become Spider-Man either. If he gets enough of a dose, he's dead. And then, of course, there's panspermia. Now, panspermia says, no, don't worry about it. No problem. It didn't happen on Earth. It happened somewhere in outer space, somewhere else in the universe, and somehow it got here, maybe on a meteorite. But of course, that's just begging the question. Okay, so fine, it didn't happen here. How did it happen wherever it supposedly happened? And that is the biggest mystery of all. Now, if we were to say, well, look, you see, science has no explanation for it. And look how complicated it is. All these parts that fit together so perfectly. And I could have gone on and on and on with the description. Must have come from God. To which I would say, as a scientist, as a mathematician, I would say, why? And how do you know? He said, well, look how complicated it is. Yeah, I know it's complicated. How do you know it didn't happen by itself? Well, I mean, demonstrate it to me. Show me scientifically why this didn't happen by itself. And what we have so far is just intuition. It's subjective. To be scientific, it has to be objective. One of the first things I discovered when I started on exploring the origin of life was that there was a website that announced a $1 million prize. It's called the Origin of Life Prize for any scientist who would figure out how, not where or when, how life started. It was suspended on October 26, 2013, after being around for 13 years. Now, the website is not there anymore. It was there when I looked at it, which was a good while ago, as you can figure out. But there are other websites that describe what was on that website. They're still around. So it's gone now. But it was operational for 13 years, $1 million offered to any scientist that could figure out how did life start. Why was it suspended? And on the website, it tells you. Here's what it said, and I quote. Over the last 13 years, since the Origin of Life Prize was first announced in Nature and Science, by the way, Nature and Science are the premier scientific journals of the day. Nature being the British and Science being the American journal. So the prize was first announced in Nature and Science. No submission has ever made it past the screening judges to higher level judges. They didn't even pass the screening judges. Why? All these scientists over 13 years, $1 million, and they can't even pass the screening judges? What happened? Why? Goes on to explain. Life origin literature continues to circumvent and ignore the key question, if not deliberately sweep it under the rug. And what is the key question? And again, I quote, how did prebiotic nature prescribe or program the first genome? The genome being the DNA, like a computer program. It encodes every detail of an organism. What kind of an organism? For example, is it an E. coli bacteria? Is it an apple tree? Is it a monarch butterfly, crocodile, human? And every detail of the organism, color, size, strength, intelligence, etc. So let's try to understand what's going on. what is the DNA? I mean, I've told you it's like a computer code, and it has all the information about what the organism is and what it's like and so on. But structurally, what is it? Turns out this is the blueprint that we talked about before, the DNA. It's a sequence of smaller molecules, which are called base pairs, and they're linked together to form a long, long chain. Now, this chain is usually wound around like a helix, and there's usually two of them, and they just go like that, like a double helix, around and around and around. And that's what the DNA consists of. It can be minimum a few thousand long, or it can even be into the billions. As it is for humans, it's over 3 billion long, into a double helix. Now, these molecules form a 4-letter alphabet, an alphabet, like a language. And that language, and it's been decoded, this language describes not only all the proteins in your body, but also when the proteins are supposed to be done and all controls of when you should produce this, when you should produce that, under what circumstances do you do it this way, under what circumstances do you do it that way, and so on and so on and so on. All the kinds of controls, data controls, the whole inside of it like a computer. If these base pairs are just random, then there's no information. It doesn't describe an organism any more than a random sequence of letters describes anything. It's meaningless. There's no information in it. So they've got to be put together in the right order in order to make sense. The letters to make sense in whatever language it's written, and the base pairs in the right order so that they describe properly an organism. Now, unlike the letters of a language, if you misspell a word, or even 2 or 3, you can usually figure out what the sentence is saying. You can recover what the word is. But in the DNA, one error doesn't necessarily cause a fatal mistake, but it usually does. Most of the time, a single error out of billions will cause the organism to be either not viable or fatal. For example, in the human genome, there are over 3 billion base pairs. One mistake, usually called a point mutation, just 1 point on the whole 3 billion plus long helix, can cause hemophilia, certain kinds of ovarian, breast, and colon cancer, sickle cell anemia, Tay-Sachs disease, cystic fibrosis, certain kinds of diabetes, Hutchinson-Gilford progeria. This is a disease in which a young person, boy, girl, young kid, has this disease, ages at an accelerated pace, and dies of old age at the age of 12 or 13. Gray hair, looks like an old man, just he's still small. One mistake. So now you understand what the problem is. The problem is you've got to get it all in the right order, and in almost all cases, you better not make a single mistake. A couple here or there, you might get away with it, but very unlikely. You might be lucky, but you've got to get almost all of them right. If you get even one wrong in the wrong place, and most of them are the wrong places, it's not going to work. The astrophysicist Fred Hoyle, and I quote, "The probability is like the chance that a tornado sweeping through a junkyard might assemble a Boeing 747. The Belgian biochemist and Nobel Prize laureate Christian René Duduve, talking about RNA. Now, RNA is the same as DNA, almost exactly the same as DNA, except that it's a single helix instead of a double one. And then there's one molecule which is different than in the DNA, but it's simpler than DNA. Talking about RNA, he called for a, quote, rejection of events with such minuscule probabilities that they may be called miracles and are not amenable to scientific inquiry. So what the scientists are saying is the probability of getting all of these things lined up just perfectly is so incredibly small, no way it could have happened. How it happened, they don't know, but they say it just doesn't make any sense. Well, okay, that fits with our intuition. Of course, think about getting 5,000 English letters, for example, all in the right order so that it reads from beginning to end without a mistake, just by chance, by spilling out a huge pot of alphabet soup on the floor. But there's a possible answer. Professor George Wald, a Nobel Prize-winning biologist, writing in the Scientific American in 1954, and I quote, "Time is in fact the hero of the plot. the time with which we have to deal with is of the order of 2 billion years. What we regard as impossible on the basis of human experience is meaningless here. Given so much time, the impossible becomes possible. The possible becomes probable, and the probable becomes virtually certain. One is only to wait. Time itself performs the miracles. So what George Wald is saying is, yeah, the probability is very, very small, but you've got a huge amount of time. According to science, 13.8 billion years. And not only that, not only do you have billions of years, he said 2 billion, scientists would say even more, but you also have a huge universe, and it could have happened anywhere. Wherever it would have happened, if it had developed into people like us or intelligent beings, they would say, why are we here? Wherever it is. So it could be just here. But that's the point. There's so much space, there's so much time, it's going to happen by chance anyway. You know what it's like? It's like a person buying a lottery ticket. It's got a 1 in a million chance of winning. Okay, you don't expect to win. But if you have 5 million people buy lottery tickets, it's fairly certain that at least one person, maybe even as many as 5, might in fact win the lottery because there's so many chances to win. Well, over here, there's also a lot of chances to win, all the way through 13.8 billion years and all the way through the entire universe, which is enormous in size. So to find out whether or not Wald had a valid objection, I decided I would have to do the calculation myself. Now, fortunately, having worked for the United States Department of Defense for 28 years as a senior cryptologic mathematician, I possessed the necessary skills to make that kind of a computation. And so I went ahead and proceeded to do so. I started with a particular virus, Phi-X174. It's a virus that infects the E. coli bacteria. Technically, it's called a bacteriophage because it infects some bacteria, but it's a virus. It's a kind of virus. The reason I picked it is because it has the smallest DNA known to mankind. The DNA is only 5,169 base pairs long, and it's a single helix instead of a double one, like RNA, so that it's half as complicated, and it's the shortest one there is. So you would think that if anything could come about by chance, surely that's got the best chance. You're not going to get a better chance with something which is more complicated. So number one, I had to calculate the probability of Phi-X174 forming all by itself. Now that's assuming that the base pairs exist already. Okay, we give them that. Okay, it's a big question, but let's just assume they're there and they're all in the same place, in the little pond. So now the question is, they're going to start linking together randomly somewhere in the universe, right? It's going to start linking together randomly, and the question is, what's the probability of getting Phi-X174? That's step number one. Step number two is it didn't have to be Phi-X174. Supposing it's a different virus, it may not be as small as Phi-X174, a little bit bigger. Well, in the universe, maybe there are some that could be viable, and they may not even exist on Earth or anywhere else, but they still might be viable. So you have to take that into account. It can't be specifically Phi-X174. With respect to the example I gave before about the lottery ticket, you're not going to ask, what's the probability of this guy, this particular person winning the lottery? What's the probability of someone in the group of people who bought tickets winning the lottery? That's the question you want to really ask. It's the same thing here. You don't want to know what's the probability of Phi-X174 forming, but what's the probability of something similar to Phi-X174 forming? And it can form anywhere in the observable universe and any time over the last 13.8 billion years. That's what we want to compute. And I did it. And what I got was a probability, which is a decimal point followed by 3,999 zeros and a 1. Now that's an incredibly small fraction. Let me try to give you an idea, because it's very hard to grasp such a small number. Give you an idea of what that means. That's like playing Russian roulette. Now, I don't know if you know what Russian roulette is. Russian roulette, a person, not you or I, but a person takes a revolver, and there are 6 chambers. He puts one bullet into one of the chambers. He spins the cylinder, and then he points it at his head and pulls the trigger. He's got a 1 in 6 chance of dying, 5 out of 6 chances of winning, depending on whether the chamber with the bullet ends up where it's going to be fired through the barrel of the gun. Only the Russians would invent something like that. What is the probability of a person playing Russian roulette 50,517 times and surviving. Would anyone in his right mind try it? If someone said that he had done it, would you believe him that he survived? You know, if you do it only 6 times, it's 2-to-1 odds that you're dead. Can you imagine 50,517 times? You know, if you wanted to write it as odds, you would have to write the word trillion, 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 333 times. Trillion, 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 333 times to 1. Those are the odds of a virus DNA similar to Phi-X174 in terms of being very small in size, coming about all by itself anywhere in the observable universe since the beginning of time. For all practical purposes, it doesn't happen. Can't happen. In fact, scientifically, the scientific approach would say we reject that possibility because the probability is too small. It's smaller than any scientific threshold set for any kind of a test of this sort. We haven't taken into account the fact that DNA by itself is totally non-functional. It's like an app without a phone, like a program without a computer. It cannot infect a cell and reproduce, or even maintain its structure and not disassemble without protection. In a cell, the cell protects it. But in a virus, it's protected by a protein sheath that protects it from the environment, holds it together, and then will enable it to infect the cell so that it can reproduce. The protein sheath for Phi-X174 consists of 192 proteins. If we were to take that into account in calculating the probability, it wouldn't be decimal point 3,999 zeros and a 1. It would be decimal point 26,588 zeros and a 1. But we didn't do that. We just did the DNA itself, and that itself shows it can't be done. And we haven't taken a whole bunch of other things into account. For example, DNA, we assumed it could be anywhere in the universe, but the fact is DNA cannot come into existence in stars. It's much too hot. It'll rip apart any molecule that I formed to be life. Organic molecules just come apart in the heat. So it can't come about in any star. It also can't form in the intergalactic media because in between the galaxies is too little atoms, particles for it to form from. They're too far apart. So a large part of the universe is already pushed away and said, no, it can't be in that. We didn't take that into account. We also didn't take into account that DNA probably could not form for the first 8.6 billion years of the universe because of gamma ray bursts, which would destroy them. In addition to that, 90% of the universe has great fluxes of radiation that would probably destroy it as well. And 24%, almost a quarter, of the universe is helium, which is totally inert and cannot form any kind of DNA or anything else for that matter. And not only that, a virus is not alive. It's a piece of DNA. It doesn't reproduce, it doesn't metabolize, it doesn't do anything until it gets into a living organism that has cells, and then if it can insert itself into the cell, it can cause havoc, as we well know. Now, we mentioned before the cell. We concentrated on the DNA, but in fact, you can work with the cell as well. Turns out the cell has a communication network inside of it called the interactome. The interactome is a communication network that connects all the different parts of the cell, the things we discussed before. How does the production machine tell somewhere else that I need this and this raw materials so that the tRNA can bring it? It's got to send a signal. So it's got to be able to transmit the signal, and at the other end, it's got to receive the signal, and so on. Signals going all over the place. This is called the Leventhal paradox of the interactome, and it appeared in an article in Protein Science in 2011, authored by, I can't remember their names right now. I will give it to you. Let's see if I can find it here. Peter Tompa and George Rose. There we go. Peter Tompa and George Rose, Protein Science 2011, the Leventhal paradox of the interactome. Now, they do part of the calculation. I finished the calculation. And what you get, instead of a decimal point 3,099 zeros and a 1, you get a decimal point 7,055 zeros, and then followed by an 8. And those are with very, very simplifying assumptions. It's much, much smaller, but they just wanted to make things easy for themselves. You know what this is the same as? Let's not even talk about Russian roulette. No one plays Russian. I'm not even sure if the Russians play Russian roulette anymore. But supposing I were to take someone out into the street and we would pick out 238 random people, the guy doesn't know them at all, and say, go ahead, guess. No cheating, everyone's watching him carefully. Guess. What's the name of that guy there? What's the name of that woman there? The name, their credit card number, the expiration date, and the security code. That is a decimal point followed by 7,055 zeros. 238 people guess all this information about them, knowing nothing to be given. Just guess, random guess, and you guess all that information. Name, credit card number, expiration date, security code. That's what that's equivalent to. And by the way, similar to the DNA, the interactome is very sensitive too. If you have errors in the interactome, it's going to wreak havoc in the organism. And errors can be caused by bacteria, by viruses, by chemicals, by age, and by mutations, radiation, for example. And it can cause things like bacillary dysentery, cholera, peptic ulcers, tuberculosis, hepatitis, polycystic kidney disease, congestive heart failure, mental retardation, cancer, cystic fibrosis, Down syndrome, Huntington's disease, muscular dystrophy. You know, just imagine if you were to rewire the inside of a car, right? So you wire the accelerator to the brake and the brake to the accelerator. One simple change, a simple interchange, you're going to wreak havoc with the car. Or think about it in an airplane. You know something? Think about the space shuttle. The space shuttle was one of the most complicated things built by mankind. It was so complicated that, believe it or not, that I heard this from the chief engineer of NASA at the time. The probability of a space shuttle on any flight having a fatal accident because of a fatal dysfunction of something inside this complicated machine was 1 in 13. 1 in 13. One chance in 13 that the astronauts getting into that space shuttle would never come back alive. You know why? Because it's so complicated. There's so many parts to it. And so many parts are absolutely necessary. Without that part working properly, it doesn't work. The whole thing goes. And the human cell, or any living cell for that matter, is far, far, far more complicated than even the space shuttle. Can you imagine if beings, human beings or animals, had a 1 in 13 chance of dying every time they went into space, whatever? 1 in 13 chance, no one would live for very long. Now we're understanding what the interactome does. One little wire going wrong can cause havoc. The authors of the Protein Science paper, Peter Tump and George Rose, writing about the spontaneous genesis of an interactome, said it couldn't happen, and I quote, within any meaningful span of time. And then they go on to say, every living cell templates the interactome of the mother cell. But you see, the mother cell had to template its interactome from the mother cell before it, and before it, and before it. Where did it start? It came together by chance? No way. The probability is much, much too small. It's telling you that it's illogical to assume that something like that could have happened. The organic chemist and molecular biologist, Alexander Graham Cairns Smith, and I quote, "But you may say, with all the time in the world, and so much world, the right combination of circumstances would happen sometime. Is it not plausible?" The answer is no. There was not enough time and not enough world. The problem with the origin of life is not a biological problem. It sounds like it should be, origin of life, biological. It's not. I mean, there are biological problems associated with it, that's true. But the real problem with the origin of life that no one's been able to solve, you know why? Because it's not a biological problem, it's a mathematical problem. And we have just shown mathematically that it's an intractable problem. It's not going to be solved. You can't get so much information together, all in one place, in the right order, in the amount of time that the universe has been here or anywhere in the universe. That's what we've demonstrated. We've done the math. We've crunched the numbers, and the numbers don't lie. As a result of this, I would imagine, George Wald, who said that time is the hero of the story, time works the miracles, he had a change of heart. 1984, he wrote in the International Journal of Quantum Chemistry, I must confess, with some shock at first to my scientific sensibilities, that mind existed always, the source and condition of physical reality. It is mind that has composed a physical universe that breeds life. But what kind of mind can this possibly be? It existed always. It's the source of all existence. Creator of the universe and life. Couldn't be life as we know it because there was nothing over there. There was nothing then. It couldn't, the life itself that exists today couldn't create itself. It had to be created by someone who designed it. And that's what George Wald is saying. He's not willing to say God did it. What he's willing to say is it was some kind of a mind, and it must have been a very impressive mind, obviously, to be able to design a universe capable of supporting life. And there's another talk completely, and in the book as well, about that subject, which is a totally different subject, and designing life, producing life, creating life to fill the universe, which is designed to have life in it. We have a name for that mind. We call it Hashem. This has been a small sampling from the book The Cosmic Puzzle, a scientific investigation into the existence of God by Harold Gans. Thank you very, very much for your attention. And join me, please, in a few days for the next talk, the second part, in a sense, of this series, which will deal with the authorship of the Torah and also who runs current events. Again, purely in a mathematical way, of course.